Hello everyone. So in previous video lecture we have started the sexual reproduction in flowering plants and have studied about flowers as sexual reproductive organ of angiosperms, parts of flower and male reproductive part of the flower. So here we will begin with the female reproductive part of a flowering plant. So the gynoecium represent the female reproductive part of a flowering plant. Gynoecium may consist of a single carpel if it is a monocarpellary and may have more than one carpel when it is multicarpellary. Uh, Sometimes pistil is the term is used in similar manner to carpel in some situation, but not in all situations. The term pistil is sometimes used to refer a single carpel or several carpels to each other. So the pistil is a collective term for the carpels when there are more than one carpels are found in a flower. So this is to note that uh, if there are more than one carpels, the carpels may be fused together to give rise a syncarpous ovary as shown here. Here all the carpels are fused to give rise a syncarpous ovary. But if they are not fused and they are free, then the ovary will be an apocarpous ovary. As you can see here, these are all the carpels which are not fused together. So these are all the carpels which are not fused together, and the ovary here is called as apocarpous ovary. Now let's see the part of the pistil. Pistil has the following three parts. First one is ovary, second one is style, and third one is a stigma. A stigma is a flat surface on which the pollen grains after pollination landed themselves and get germinates. Style is found just below the stigma. It is a tube-like structure that connects the stigma with the ovary, and the ovary is a swollen part in which the ovules are produced. Inside the ovary, there is a hollow space which is called as ovarian cavity. This ovarian cavity can be divided into several chambers by several septum, and each chamber is called as locule. The placenta is located inside the ovarian cavity, and I think you have already read about the placentation in class 9, class 11. So I'm not going to deep in for them. It will be very long topic. So in the locules, placenta will be there on which the ovules are arises. Uh, there may be there will be a single ovule in a locule in a ovary, or as like in wheat, paddy, mango, or it can be many ovules in a single ovary, as like in papaya, watermelon, orchids. So on the basis of structures, ovule can be of basically six types. We will study in next slides. All the ovules have similar parts. So to understand a brief idea about the structure of ovary, we will take an example of an anatropous ovule, and so, which have following parts. First one is the funiculus. Funiculus is a stack by which the body of ovule is attached with the placenta. Here would be the placenta by which the funicle attaches the body of ovule. Uh, integument is the protective envelope that surrounds the whole body of the an ovule. That is the nucleus. Nucleus is surrounded uh, by the integuments. Integuments is a can be a single layer or can be of two layer thick. If this is a single layered thick, then it is called as unitegmic, where say a single integument is found. And for example, compositive family, members of compositive family has only single layer integument. But if there are two integuments, the condition is called as bitegmic, and this is very common in the members of Papilionaceae family. This is to note that I forgot to mention that the point of attachment of funicle with the body is called hilum. So there would be the hilum, where the funicle attaches with the body of the ovule, and sometimes the funicle extends to the base of the base of the ovule, like here, and get fuses with the integument to give rise a ridge-like structure, and it forms the raphe. So here you can see that this is the raphe, and that is formed due to the fusion of the uh, funiculus with the integument, and it is common in anatropous ovules. So if you will see a seed coat, there will be a line or ridge is visible, that is the raphe. As you know that ovule will make a seed in future. The third most important part is the nucellus, and the integument includes the large parenchymatous tissue within them, and this is the nucellus. It is very important part, as a cell of the nucellus will act as the uh, megaspore mother cell. You will learn them about the future, not here I am going to confuse, otherwise you will be confused. So this is a central part of an ovule, and it encloses the embryo sac. uh, if you will see here, when the integument surrounds the nucellus, there are two portions, one here and one here. The integument surrounds the nucellus all around, but at one single place it do not surrounds and keep their simple tip open, and this open tip or a small opening is the apex of the integument, and it is termed as micropyle, while opposite of the micropyle is the portion which is called as calaza. The spelling is CHALAZA, so sometimes it is also studied or pronunciated like chalaja, but it is not the chalaza, it is calaza. Embryo sac is situated in the center of the nucellus. It is the female gametophyte, and which develops from a functional megaspore. We will study these terms later. But there are more, as I mentioned earlier, that there are mostly 6 types of ovules, and we have seen a structure of anatropous ovule. Anat- uh, so there are mostly 6 types of ovule: orthotropous, anatropous, hemianatropous, uh, arsenotropous, compylotropous, and amphitropous ovules. So there are 6 basic types of ovules. So in an orthotropous, or which is also called as atropous ovule, if you will see, the body of the ovule is completely straight. Actually, the difference in the ovules is due to the arrangement of the micropyle and the scalaza, mostly with the funiculus. So if you will see here, the funiculus, micropyle here, and scalaza all found in a straight line and make us 180 degree of angle. So the body of ovule in atropous ovule is erect or straight, where the hilum, calaza, and the micropyle lie in a straight line. You can see that this would be the portion of hilum where funicle will attach with the body of the ovule, and this is a very ordinary type of ovule found in polygonal plants, etc. Second one is an anatropous ovule. You have seen a detailed structure of anatropous ovule here. So in the anatropous ovule, the body of the ovule becomes completely inverted due to development when the funicle mm, uh, develops very much. So it gets somewhat inverted, and in this situation, the micropyle comes to very near to the hilum. here the micropyle and this is the hilum, so they come to the very close to each other. In the it is found in the members of gamopetalic mostly. Third one is hemid anatropous or hemitropous ovule. The body of the ovule is placed transversely at a right angle to the funicle. This would be the funicle, and this funicle, and um, this is the body of the ovule. So you will see that this will be a 90-degree of angle. So this is a very uh, important to note that the funicle and the body of the ovule makes a right angles here in this type of ovule. And this is also to note that the micropyle and the callasa found in a straight line in such type of ovules. This is the ovule found in the rene pedunculus, for example. Four type of ovules are campylotropous ovule. Campylo, as the word suggests, means curved. So the body of ovule in a campylotropous ovule is curved or bent round, so that the micropyle and the callasa do not lie in the same straight line. Here you will see that the micropyle and here will be the callasa, so they are not in the straight line. They will be somewhat curved. And such type of ovules are found in leguminosae. The fifth one is the amphitropous ovule. The curvature of the ovule here is very much uh, pronounced, and the embryo sac. If you see that it is somewhat like the campylotropous, but here embryo sac also get the curve. This type of ovule is found in the Alice Macy and the Butomaceae families. Sarcinotropous ovule is a very special kind of ovule. Here the nucleus and the axis of the ovule are in the same line in the beginning, due to the rapid growth of the funiculus or the axis, the ovule become anatropous. And this is to note that here the funicle um, actually surrounds the whole ovule from around. So the curvature continues further, and the micropyle again points the upward. So due to the surrounding of the ovules or the bending of the ovule, the micropyle come to the upper portion part. So such type of ovules are common in the opuntia, which is a desert plant. Now let's see about the gametogenesis in flowering plant. As we are studying the uh, pre-fertilization event, so there are mostly two events are common. First one is the gametogenesis, and second one is the gamete transfer. So let's begin with the gametogenesis. In flowering plants, we can divide the whole process of formation of gametes in two parts. First one is the sporogenesis, that is formation of spores, and second one is the gametogenesis, that is for complete formation or development of the gametes. We will divide the gametogenesis in two parts. First, in for the male plants, and the second male parts, not the male plants. First for the male parts, and the second for the female parts. In male parts, the sporogenesis will be microspore, as the microspore will be formed here, which is also called as pollen grain. I think you have heard the word. And second step will be microgametogenesis, that is the development of the male gamete in the male part of the plant. And in female parts, there will be again a megasporogenesis, that is the formation or development of a embryo sac, and inside the embryo sac, a megagamete, that is a female gamete or egg, will be developed, and the event will be called as megagametogenesis. So let's begin with the microsporogenesis. So first of all, microsporogenesis can be defined as the formation of microspores, that is pollen grains, or the uh, male gametophyte inside an microsporangia, uh, or it is also termed as pollen sacs of a seed plants from the microsporocytes or pollen mother cells or microspore mother cells. These are the terms mostly used interchangeably. Cells of the so let's see how the microsporogenesis take place. You have seen a structure of mature anther in previous video lecture. If you have forgot this, again revise it from there. This is the benefit of video class. You can go to the video and open the video to again revise the chapters. It's like classroom. This is not possible in ordinary classroom. Yeah, of course both of them have their own benefits. So if you see there, in the amateur anther there were a type of cells called sporogenous cells. I have remembered these cells and were enveloped within the anther walls. So cells of these sporogenous tissue are capable to give rise to pollen tetrads. Pollen tetrad. What do you mean by pollen tetrad? Pollen tetrad means a cluster of 4 pollen grains or 4 pollens, and hence they act as the pollen mother cells. It is PMC. It is also write as the small MC, that mean microspore mother cell. So it is also termed as microspore mother cell. So it is write as small MC. Always remember, if in the examination ask the question that write about the microsporogenesis, don't write the capital M, capital M and capital C, because it will represent the megaspore mother cell. So small MC, that is microspore mother cell, from will be formed from the sporogenous tissue. And actually the sporogenous tissue will act as the microspore mother cell. The nuclei of these pollen mother cell or microspore mother cell undergoes meiosis to form a cluster of 4 nuclei. And I think you should take a pause to uh, remind what would be the ploidy of the pollen mother cell and what would be the ploidy of the these pollen tetrad that will be formed after the meiosis in the pollen mother cell. Yeah, of course, you are right. The nuclei of these pollen mother cell undergo meiosis to form cluster of 4 nuclei. So the pollen mother cell will be diploid in ploidy, and when there are two continuous division take place, meiosis first and meiosis second in the pollen mother cells. So during meiosis first there is a reductional division will take place in the nucleus of pollen mother cell. After this, 2 nuclei will be formed, and these nuclei will have n number of chromosome. This n number of chromosome, uh, and they will be haploid and will have half sets of chromosome in them. So in first 2 nuclei which are formed haploid, now these nuclei will undergo meiosis second, which is equational division, and the haploid nuclei will form again. Each nuclei are haploid, will have n number of chromosome. So the nuclei of this pollen mother cell undergo meiosis and form cluster of 4 nuclei, and these cluster of nuclei are termed as pollen tetrad. As soon as they develop a cell wall around them, these nuclei will develop a cell wall around them and will consider as the pollen tetrad, as they are not separated to each other. Since they are not separated to each other, a specific arrangement of pollen grains in the tetrad is seen, and this the structure of this tetrad is affected by the cytokinesis during the meiosis. And the cytokinesis in them can be of two types, either simultaneous, like in the uh, dicot plants, in the condition of dicotyledon plant, or successive type, like in the monocot plant. In the simultaneous type, cytokinesis occur only at the end of meiosis second. Here you will see that uh, the nuclei is dividing. Here 2 nuclei are formed, then again nuclei are formed, and the cell wall was not developed here. You can see that the cell wall is not developing, and when the complete meiosis, that is meiosis second, is also completed, a cell wall is formed. So in this situation, generally tetrahedral type of pollen tetrad is formed, while in the successive type, uh, once at the end of meiosis, the cytokinesis occur twice. Once at the end of meiosis first, forming two cells, and then again, like you see that after meiosis first, cell wall formed and two cells are formed here separately. Then again meiosis 2 take place in both the cells, and the cell wall again forms, that is cytokinesis again happen after the meiosis second. So the cytokinesis take place one after the one, first after meiosis first and second after the meiosis second. So in such type of pollen tetrad, generally isobilateral arrangement of tetrad is shown, and it is generally found in the monocot plants. So, what can be the arrangement of pollens in pollen tetrads? In different species of arrangement of pollens in a tetrad may be different. It can be linear, where one pollen found above the other to giving rise as linear, straight line structure. Second can be T-shaped, like here you can see there is a T-shaped structure. Uh, these two are making the head of the T, and these are the tail of the T. So the second one is decussate, you can see here, and third one is by isobilateral. All the are the equally and bilateral division. Tetrahed or can be tetrahedral. Pollen tetrads of most of the plants are tetrahedral in arrangement. This is to note that in mostly dicotyledonous plants form the tetrahedral pollen tetrads. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspores or pollen grains are formed that are released with the dehiscence of the anther. So, as the anther matures, uh, it dehydrate, and the microspore dissociate from each other and develop uh, into pollen grains. The process of anther opening, that is the essence of anther, was followed in different species by means of light and electron microscopical techniques to study how the dehiscence of the anther take place. So when the experiment was taken place, a sequence of the anther opening mechanism was outlined, that contain following stages, that is first expansion of epidermis and endothelial cells, and deposition of U-shaped wall thickening inside the latter. Here in that situation, the of first one, all the microspores which were earlier divided by the septums or cells here, uh, get fused to give rise only two microsporangium. Then the enzymatic opening of the septum between two locules take place, and the mechanical rupture of the septum take place. After this rupture of septum, the opening of the stomium, this is the line in the epidermis. Stomium take place by the force of highly turgescent epidermis and endothelium, and outward bending of the locule walls by the centrifugal force of the dehydrating epidermis and endothelium. So anther dehisces, and the mature pollen grains releases from here. So pollen is so now let's look how a pollen look like. So if you see, the pollen grains are generally spherical. Of course you can see a pollen grain with the help of a microscope. It is in the syllabus of your class 12, that you will make a slide of germinating pollen tube. You will uh, collect the pollen grain of sunflower, mainly of hibiscus, and then you will germinate it with the help of a culture or medium containing sucrose and several other salts. So You can see the pollen grains with the help of a microscope, and you will find that most of the pollen grains are generally spherical in structure. Now you can see here, but some can be elongated to oval structure, but mostly are spherical in structure. Architecture of ovule is very interesting. Mostly size, shape, color, designs of pollen varies species to species. And if anyone asks you about the diameter of a pollen grain, this is not fixed, but approximately 25 to 50 micrometers can be diameter of a pollen grain, and as it is mentioned in the NCRT book. Uh, a pollen is provided with two coats. The external surface, external body of the pollen tube. The pollen grain is contains of two coats. First one is exine. It is also called as extine, and it is outer tough cutinized layer called layer. The exine is often sculptured. Actually, the sculpturing you are seeing here, this is ridges or this is tufts or the spine-like projection, all are in the exine layer of the pollen grains. So, is provided. The exine is actually made up of sporopollenin, which is highly resistant to temperature and acids and alkalis. This is the first organic matter or the organic matter yet known which is highly resistant, and this is the reason you can find a pollen of a plant. It will not be viable, of course, but you can find pollen grain of a plant after thousand or thousand years if it is not taken by any animal, but it can be buried in soil and it can be extracted from there. And hence the study of the pollen grain is also a very important branch of fossil study. That, and the exine is the layer that that will help in the preservation of the pollen in any extreme condition very easily, and the sporopollen is the matter due to which this can be possible. The intine is the second layer of the external wall of the pollen. The inner wall of the pollen grain is called intine, and it is very thin and continuous layer made up of cellulose and pectin. In the entine layer, in exine layer, you can see that there are some places where the exine is absent. This layer is called, these portions are called as germ pool, and this is the reason from where the pollen tube in future can be germinate when they will fall on a stigma after the pollen pistil interaction. The germ pool, the pollen tube will be germinated from these germ pores like this. Thus, but there is no any such type of differences or such type of gaps are found in the intine layer. It is completely uh, continuous layer and made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. So here is the last layer, which is the plasma membrane. So first one is exine, second was intine, and the last one is the plasma membrane. Uh, if we see the section of a pollen grain, we can clearly see two types of cells here. First one is vegetative cell and second one is generative cell. The formation of the vegetative cell and generative cells take place after the maturation of the pollen grain. Firstly, when pollen grains are not matured, there will be only vacuole and nucleus shown, and after the division in the nucleus, uh, an asymmetrical spindle take place, spindle formation take place, and due to this asymmetrical spindle formation, the division of the nucleus will be an asymmetrical division where a large cell is formed and a small cell is formed. The large cell is called as vegetative cell, while the small cell is called as generative cell. The vegetative cell has abundant food reserve provided to give the generative cell, and it has a large nucleus. You can see the difference in the size of the nucleus of the vegetative cell and generative cell. It is a large nucleus, shaped nucleus, and the nucleus of the vegetative cell is irregularly shaped. while the generative cell is very small and it generally floats in the cytoplasm of the vegetative cell. It is spindle shaped. You can see it is a spindle shape and have dense cytoplasm and then prominent nucleus, as it will uh, divide into a gamete, two gametes, and so it will act as a gamete. So for acting as a gamete, this is very important that they will have proper cytoplasm and a proper nucleus. Next topic is microgametogenesis. So, what is microgametogenesis? As we have earlier mentioned that there are two portion will be, first one is microsporogenesis and second one is the microgametogenesis. So, microgametogenesis is the formation of gametes, microgametes, male gametes, uh, that will take place inside a pollen grain. uh, it take place by the mitotic division in the generative cell. When a mitotic division, that is mitosis take place in a generative cell, the formation of microgametes is take place. In over 60% of angiosperms, pollen grains are shed at the two cell stage, that is a generative cell and vegetative cells. And but in 40% of the plants, the pollen grains shed in a three cell stage. That is, there will be two gametes have been formed from the generative cell and one vegetative cell. Vegetative cell provide the nutrition to the gametes, that is generative cells. But this is not that there will be only two cells or only one gamete and one vegetative cell in the microgametes of 60% plant. The generative cells of these plants will also divide, but when it will reaches to the stigma, it will shed to landed to the stigma, and after the pollen pistil reaction, when the pollen tube germinates uh, from the pollen. At the same time, the generative cell of these 60% angiospermic plants divide to give rise to gametes. So this is the gametogenesis, a microgametogenesis in the male of the flowering plants. So let's move to the megasporogenesis, that is the development of uh, megaspore, that will develop into embryo sac from a from a megaspore mother cell. The megaspore is also termed as the female gametophyte. Megagametogenesis, as we have already seen, that it will also take place in two steps. First one is megasporogenesis and second one will be the megagametogenesis. So megasporogenesis is the formation of megaspore, that is a female gametophyte from the megaspore mother cell. Uh, so if you have seen a structure of ovule, you have just seen the structure of ovule, you can pause the video and go back side to see the structure of ovule again. This is in your hand now. During the megasporogenesis, one hypodermal cell of the nucellus, one hypodermal cell of the nucellus, uh, that is generally of micropylar side, becomes differentiated from the others by its bigger size, dense cytoplasm, and conspicuous nucleus, and which is this is termed as the archisporial cell. Once let's take you back here. This is the nucleus, and this is the micropyle. So any cell here, if there is not, we will considering the condition when there is no any embryo sac. So there will be only cells, and only cells. So one cell of this nucellus will be modified, will be differentiated, and will be in bigger in size, dense cytoplasm, and the nucleus will be of this cell will also conspicuous. And in such condition, this cell will called as archisporial cell. The archisporial cell function as megaspore mother cell, and the megaspore mother cell is diploid in, that is 2n, and will undergo meiosis first and then in meiosis second to form 4 haploid N megaspore. N megaspore. N means haploid megaspores. Uh, Haploid megaspores. So you can see that this would be a micro archisporial cell here, and this is archisporial cell, and this archisporial cell first divided by meiosis first to give rise a megaspore dyad. First one megaspore and second megaspore, and this archisporial cell will be n, sorry, 2n. It will be 2n. So these megaspore dyad will contain two cells having only n number of chromosomes, that is haploid. Again, these megaspore cells of the dyad will undergo meiosis second to give rise these four my megaspores cells or megaspores, uh, and the condition will be called again as megaspore tetrad. There was the macrospore tetrad, is pollen tetrad, and here it is megaspore tetrad. The megaspore mother cell was diploid, so the megaspores will be haploid in condition. Uh, the megaspore tetrads mostly are arranged in linearly, that is one above the other, and most of the cases it is linear in arrangement. This is to note that after these four megaspore tetrads, three gets degenerated and only one remain functional. The functional megaspore gets nutrition from nucleus and enlarges to serves as the function of female gametophyte. Uh, so if you will see that this is the special kind of development of embryo sac that is found in, that is very common in angiosperm. It's very common type of embryo sac development, and as here three megaspores get degenerated and only one remain functional, and the embryo sac development take place from these single megaspore. So it is generally found in ordinary type or normal type of, or also what can we say, that polygonum type of embryo sac. uh, this is very common, and it is called as monosporic type of embryo sac development. But it can be, there are several types of other micro embryo sac development, like bisporic, tetrasporic, and so many. But you will study, and only the megasporogenesis of monosporic type, where a single megaspore is functional and will develop into a mature embryo sac. So now let's see a look about the megagametogenesis. This is very important topic and very interesting topic. Uh, Inside the female gametophyte that was developed as after the enlarging itself from the megaspore. When the megaspore enlarges itself by taking the nucleus, the female gametophyte is developed, and this female gametophyte has a single nucleus, and this single nucleus uh, undergo a mitotic division to give rise to nucleus. Among these two nucleus, one has migrated towards the calazoline and one towards the micropylarin. That is like this. If this is a condition, here a nucleus, this nucleus has divided, and one nucleus migrated towards the calazoline, and second one is divided towards the uh, micropylarin at the both ends. So there are now two nucleus at the both ends, sorry, single nucleus at the both ends, and now these nucleus will divide successively two times. One, when one nucleus divide, two nucleus are formed, and similarly at the second end two nucleus are formed, and when these two nucleus divides, again four nuclei are formed. Among these nuclei, one from each end migrates toward the center again, and when it migrates toward the center, there will be two nuclei at the center of the embryo sac or the female gametophyte. These two nuclei fuses with each other and forms a polar nuclei, and after the development of the wall around them form the central cell having two nuclei, and it is in future will be represented as 2n. While the other ends will have um, three cells at each end, three cells at callosal end, and second three cells at the micropylar end. These cells will again form the special kind of structures, like at the callosal end these three cells fuses with each other and constitute to form antipodals, that is antipodal cells, while at the micropylar end the among the three cells two develops as the synergids, while a central cell is developed the female gamete, that is egg. So three cells are grouped together at the micropylar end and constitute the egg apparatus. This condition is called as egg apparatus. And the egg apparatus in turn consists of two synergids and one egg cell. While at the callosal end, these three cells are termed as antipodal cells. Uh, Note there mm, take a note please that the synergids have there are some special cellular techniques at the micropylar end, and these are called as filiform apparatus. These are very important structure, however their function is not completely known, but it is assumed that they play an important role in guiding the pollen tubes into the synergids. So uh, these are the filiform apparatus. So you will see there are three cells at the callosal end forming antipodals, two cells at the central of the embryo sac forming the polar nuclei or the central cell, and three cells in which two are synergies and one is egg, that is female gamete, at the micropylar end. So if you count that, there will be 3, 6 and 2, 8 nuclei uh, in the embryo sac, and but the cell will be 3 at antipodals, 3 at micropylar end including an egg, and one at the central of center of the embryo cell, as these nuclei have been fused and cytokinesis has, that is the formation of cell wall around them, has been taken place. So this will count as an only one single cell. So there will be a 7 cell structure in a um, in a embryo sac will be take place. So I am finishing here for today. In next lecture, we will continue from the pollination, that is the gamete transfer event of pre-fertilization event. Thank you for your patience listening.